I'm Paula Desplats. I'm assistant professor in the departments of neuroscience and pathology. My main focus is trying to understand molecular mechanisms of neurodegeneration. And I'm very interested in understanding how the genes are regulated, become altered, and contribute to pathology of Parkinson's, Alzheimer's, HIV dementia. Epigenetics are all those mechanisms that will model the chromatin structure and will turn genes on or off. And we are mainly interested in DNA methylation changes. We started doing genome-wide profiling, trying to see these DNA methylation marks across the brain, comparing brains from controlled, healthy patients and brains from patients with Alzheimer's. So we compare post-mortem brain samples, and we look in the frontal cortex DNA, and we found is whole changes in DNA methylation. And indeed, we were looking for changes in genes that regulate inflammation, because inflammation is a very important component of pathology. But incidentally, we hit many genes of the circadian clock, to our surprise. DNA methylation is very cyclic and also changes throughout the day. And these cycles of methylation were completely shifting in early AD patients. There is a behavioral component that is called sundowning, and that means that the patient, when the sun goes down, becomes very agitated and confused and aggressive. And that then evolves later into changing in the sleep cycle to a point that the patient will be wandering around at 2 a.m. in the morning, and that is so disruptive for the family living. It's one of the major factors for placing the patients in the institution. So you can imagine that if we have really a better understanding on how this clock became deregulated, and we can try to intervene early enough, we can maybe hold these patients with their families. And that's the most precious thing that we can do.